Nothing? Okay, so today we're going to be looking at the other side of the immune response. It's called the adaptive immunity. This is not something you're born with. This is something that your body basically gets trained as you get exposed to pathogen. And it's pretty important for a couple reasons. One, there is some evidence that if you grow up in too clean of environment, your immune system actually will not train itself very well. And there's a chance you could be subject or susceptible to allergies and other autoimmune disorders. So that's kind of an interesting, it's called the hygiene hypothesis. If you've ever heard of that, you can look it up. The other side of this, though, is even though the adaptive is kind of a slower response, I think we talked about this last time, right? Did I tell you some of the qualities of innate versus adaptive? What were some of the qualities of the innate response? It's a little slower. Did you say stronger? In what sense? Because it's not very directed, though, right? Right. Yeah, well, I mean, so strong in the sense that it probably covers a lot, right? Like the moat around a castle is very general, non-specific, but hopefully is a pretty fast response to most things that try to infect us so that we can get a start on fighting the infection off, right? What about adaptive? Since it's going to involve things like antibody production and recruiting B and T cells, that's going to take time. And I don't know, I forget if the slides talk about this, but it also takes time to generate B cells and T cells that are specific to whatever we're trying to fight off. So the responses that we do see in here are very specific for what their targets are, and that takes time, up to weeks, to get the perfect kind of response. Well, I shouldn't say perfect, but a directed kind of response to whatever we're being infected with. Okay. So these things are going to work hand in hand. And I did list both innate and adaptive things on here. So today we're going to focus on things like antibodies, B and T cells. But you have to realize there's cells involved in these things as well as proteins involved in these processes that are going to help. Okay. So what does our immune system actually respond to? It really is just antigens. So antigen is short for antibody generating. It's where they cut the term from. These are mainly proteins that are going to be found on foreign compounds, foreign cells. It's not always proteins. They can also respond to things like the lipopolysaccharides. Sometimes they can respond to RNA or DNA. I mentioned the toll-like receptors last time. So they can be specific for certain things. But generally what our body looks for are small molecules that are processed from these larger molecules. So you know that bacterial cells are a complex mixture of things. What happens in our body, or what can tend to happen, is that parts of this, maybe it's proteins sticking off of a cell, so they can recognize gram-positive or gram-negative cell wall components. Or sometimes these cells called APCs, antigen presenting cells, like macrophages. Do you remember what macrophages, how we describe macrophages? It's really me, it's my hearing. You guys say something, but then you don't repeat it, and I'm sure it's the right answer. Right, so they're known for a lot of things. Phagocytosis was one of the major things they do. So a lot of these antigen presenting cells, like macrophages, dendritic cells, will gobble up a bacteria, basically digest them, and then present those particles, a lot of the time, again, proteins or short proteins, to the B and T cells. So they're not always responding to the entire organism, if you will, but just parts of the organism that are then seen as foreign. And that's important because sometimes these organisms can change. We talked about mutations, antigenic drift, or antigenic variation. If those small things change, then our immune systems won't be able to recognize them as well. And so that could affect how they respond to the organism. So most of the time, just keep in mind that our immune system is looking at really small areas of proteins or sugars or things like that and seeing those as being different or foreign. What kind of responses do we see? There is a humoral component, and the way I remember, humoral is stuff that's dissolved in our plasma. So if you see that word, just think soluble compounds. And a lot of times these are proteins. So today we're going to talk about what antibodies are and what they do. There's also the complement proteins, which are, I think it was like 30 or so different types of proteins that float around in your body, and they can spontaneously recognize things. We talked about, did we talk about mucin and defensins in this class? I forget if it was this class or a different class, but there's other proteins like lysozyme. Those things can fight generally against things. So I put these in parentheses because they're kind of innate, right? We're not really talking about them today. Cell mediated are going to be mainly the B and T cells. So there's two types of T cells, cytotoxic T cells and helper T cells. You generally want to keep track of what these do. To me, it's a little easy. I think cytotoxic, what does that sound like? Good, bad? Like attacking, right? Toxic, bad. So not bad for us, but cytotoxic T cells are the ones that go out and attack. They can actually kill off cells like virally infected cells. And so they're the ones that have a lot of ammunition where they can go and fight things off. Helper T cells are actually going to help B cells and other cells make antibodies or secrete chemicals, signaling chemicals, which are then going to recruit other cells to the site of infection. So this kind of shows you the overall parts of these. So B cells are going to be the ones that make antibodies. So you want to remember that. And then T cells can do a couple of different things. And we're going to talk about antibodies and antibody structure as well. So B cells, you want to think antibody production. And T cells are going to either be cytotoxic cells or helper cells. And we really can't get into the nitty-gritty of all these things because that's basically why I teach the other class, the immunology class. That's where we look at the nitty-gritty of how these things work. But I'm going to try to give you kind of an overview of what they're doing and why they're important. The take-home message, though, is we need all of these things together to work. We can't really be missing any part of either the innate or adaptive immunity without showing some sort of an immunodeficiency. So all of these cells and proteins have a key role to play at some point. Okay, so what kind of happens here is when we get infected by things, the cells or proteins or a combination of these are going to recognize the foreignness of these compounds, whatever they are. That term is called antigenicity. So this is how well something elicits an immune response. So our immune system is trained not to recognize self proteins, for example, but to recognize foreign compounds, which they've never seen before. In fact, our body spends a huge amount of time and energy creating new B and T cells in the hopes that they're going to be able to find any of these types of foreign compounds. So I'm just wanting to remember my question. Okay, good. So they're going to be recognized, and then, for example, these antigen presenting cells can digest them. This is just one example of how this happens. They can digest them and then present them to the B and T cells. So there's a lot of communication that's going on in this process as well. And then what the B and T cells do is they go through this differentiation where they will then respond in their way to that infection. A lot of times that involves things like T cells actually going back to the infection site. It's called homing. You don't need to know that term. But they actually can be directed back to the infection site. That kind of makes sense. You want the cells that are going to fight the infection to go to the infection, not just float around randomly. So it's pretty cool. So T cells can go and attack, B cells can go and make antibody if they're specific for that. And then, I mean, that's kind of the whole goal. We want those cells to work together to remove the foreign body, whatever they are, okay? So the best ones are proteins, some of the strongest antigenic ability. These other ones, like nucleic acids and sugars, they don't always work the best. On Friday, we'll talk about vaccination. Some vaccines don't elicit a very good response, so you actually have to either mix them with other compounds, which set that off, or you have to, well, you mix them or you change them in such a way to increase the chances of having a good immune response. If you get a weak response, if our immune system doesn't respond well, or you don't have both B and T cells responding, then you don't generate a very good memory of that infection. And we'll come back to that, but that's another part of the adaptive immunity that's really, really important. How are we doing so far? So there are specific cells that are going to be activated by other cells, typically like APCs. And then those B and T cells are going to be recruited to go do their thing to fight off the infection. Yeah, so far so good. Okay. And we'll talk about vaccinations on Friday, but basically this is going to be a way where we use foreign particles to train up our immune system to respond to that. We could talk about the pros and cons of these later on, okay? We'll save that for next time. So here's my first question for you then. Antibodies, you probably heard of them. I got to get the right question. So my question for you is, what do antibodies do? Oh, in your body. There you go. Spell check failed me. So you already know I told you they're proteins. Let's see if you've heard of them. And if you get it wrong, you can blame it on my spelling, I guess. Everybody? Well, most of you. So our antibodies don't attack our own cells. I kind of just put these in here as distractors because they don't really have anything to do with antibodies. So the last one is the correct one. They're going to be attacking antigen. That's right. Okay, very good. And where do antibodies come from? What? B cells, right? B cells. You also hear the word plasma cells. Another term they might use is a plasma cell. Plasma cell is basically just a B cell that is producing antibody. So B cells can be at different stages. So they can just be sitting there, not activated, not doing anything, or they could be plasma cells secreting antibody, or they could be memory cells. So they can create a memory of whatever you're infected with and try to, the next time I'm ready for it. The fancy name for antibodies is immunoglobulins, or IG. You ever heard of anybody having IG therapy? They're receiving immunoglobulins from a donor, typically. Okay. Kind of a cool little structure. I don't know if you've ever seen it before. It's made up of 4 different proteins. They all come together and they have antigen binding sites. So on one end, on this end over here, they're going to be specific for binding to whatever antigen they're binding to. So typically a foreign antigen. On the other side is what's called the constant region. That constant region is the region that's going to be recognized by other cells. whether it's the B and T cells can recognize them or other things like complement. So there's other proteins basically that can interact with these constant regions. What's cool about antibodies is there's a wide variety of them. You don't need to memorize the classes for this class, okay? If you want to do that, you can take any knowledge. But what's kind of cool is we have a wide variety. So B cells have the ability to what's called switch between these different classes of antibodies, and they all have different characteristics depending on either where they are or what they're doing. And I'll just give you a couple of examples. So for example, IgA is very good at crossing epithelial sheets. So we see that produced in our mucus and in our gastrointestinal tract. And I think it's IgA is also the one that's passed through milk reversing. Correct me if I'm wrong, but I think that's the one as well. So it's got very specific characteristics which help it do its job basically. And then IgEs, that's the one that's almost always involved in allergies, allergic reactions, which we won't talk about in this class. But it's very specialized to binding to certain cells to start those allergic responses. Nobody has allergies in here, right? So you have no idea what's going on. This just gives you some kind of information on those. You don't need to memorize them, thankfully, because there's a lot of information there. You could take note about what I just said, though. They have different characteristics. So that kind of makes sense, right? The more variety you have in your ammunition, if you will, then maybe the better you can fight off different diseases, foreign antigens, whatever they are. And this gives you some pictures. So let's see. We have, this IgA, that's the IgA structure, and IgM is just another typical antibody. So you can see some pretty varied structures. What do these guys do? They basically do everything. So antibodies can bind to antigen. They can bind to toxins. They can bind to viral proteins. They can help with macrophages taking up foreign compounds. I think I mentioned that last time, where macrophages can recognize complement and antibody structures, and that helps the macrophages process these foreign antigens, wherever they are. So there's all sorts of different things that these antibodies can do. So it's talking about primary antibody response, secondary response. The point here, though, is that part of the process when B cells are activated to respond to a foreign antigen, some of those B cells become memory B cells, and they can actually they essentially mutate to become even better at recognizing that infection, whatever it was. So the first time you get exposed to something, what happens? You get the innate response, right? I'm sorry, these aren't all innate. You get the innate response first. That recruits in the cell-mediated adaptive response, B and T cells, right? But interestingly enough, the amount of antibody that's produced and the quality, really, of the antibodies that's produced first are not that great. They might not bind to the antigen as well, or they don't bind very well at all. You don't get a lot of them. But the second time you are exposed to something, we see a much different response, as long as you've seen it before. We actually see a stronger response. You make more antibody, and it binds better. It binds better to the antigen. So that's part of this memory system, is that the B cells and T cell, no, it's mainly B cells, can continue to improve on how they respond to something. And in general, the better the response, the better the outcomes. You can basically fight these things off pretty quickly. Isn't that interesting? Is that a good thing? Yeah, it is. You want your immune system to be able to train itself to fight these things off and fight them quicker and better if they can. And that's part of vaccination. We'll talk about that on Friday. part of the idea of vaccinating yourself is that you're training your immune system to see these things before you're exposed. And the next time, so the second time, if you ever get exposed to it, then your immune system is ready for that. So we can talk about that later on. And that's because a lot of the antibody diversity, since they're proteins, they all come from genes. So our cells have an ability to recombine their genes to make even better antibody. I think this is cool. And I don't know, I don't remember the number, but I think our bodies spend every day they make something like 1 million new B cells. And all these B cells, it could be more than that. All these B cells, they're just there on the chance that they can bind to foreign antigen. So it seems really weird and random that we're just constantly making these B cells with the hopes that they will bind to foreign particles. It's kind of crazy how much energy our body does. I don't know, did the book talk about any of that? How many B cells we make? Huh? I don't know. Okay. Yeah, it's kind of a kind of an interesting thing that our bodies just continually generate these things. If they don't bind to antigen, then these B cells are destroyed. They just get recycled. So it seems like a waste, but the idea is to try to have the B and T cells there before we get infected so that they have the ability to bind to these things and respond. Okay, link between humoral and cell mediated. So T cells, both B and T cells start in the bone marrow. Right? We talked about that last time. So we make pretty much all the cells in the bone marrow. T cells go to the thymus to basically mature. So the thymus is really, really important for T cell development. That's also where these cells are trained to not bind to self cells, self proteins. So that's another part of our immune system is that there's a whole training program where we get rid of B and T cells, for example, that attack our own cells. Do you know what those kind of diseases are called? Autoimmunity. So autoimmune diseases are any time when your body, for whatever reason, mistakes a self antigen for a foreign antigen and starts attacking it. So you might have patients that have this lupus and now I can't even, there's a whole bunch of them. You can look them up. But I can't think of any other ones right now. Oh, MS, right? Multiple sclerosis. That's another one where they actually attack your neurons or the sheaths around your neuron like that. T cells, this is going in a little deep here. T cells will actually bind to what these things are called MHC compounds. So I don't think I'm going to test you that much on MHCs because there's a lot of information here. It's a lot to take in just one lecture. If you ever get into an area where you're doing a lot of transplantations, so transplantation surgeries, those kind of things, when they're trying to match up the donor and recipient people, they actually have to look at these MHC proteins to make sure they match. Because these proteins are really, really important in the presentation process. So in presenting the antigens to the cells. So I think just look these over. These are just important. You need to know that they're important for the T cells to recognize the correct antigen, okay? So just try to remember that about the MHC proteins. And this picture here shows the process of how here it's a viral protein is being cut up, and then the MHC molecules are going to then display that to the T cell receptors. So I can't really do justice to all these processes in just one day. So really, if you want to see the nitty gritty, you're going to have to go in and study it a lot more. But this T cell receptor is the one that's recognizing the MHCs. Does it show that picture? Yeah, it does right here. So here's the interaction between T cells and the antigen presenting cells. And so what they're showing is the T cell has receptors that will bind to the antigen presenting receptors, which are the MHC proteins. Okay. So I think here what you should do is just take a step back and go, wow, this is really complex. How the immune system has all these functioning parts. And there's a lot of communication that goes on. And that communication has to be correct. You can't just have the immune system doing things willy-nilly, because then you're going to have hypersensitivity reactions, that's like allergic reactions, or autoimmune reactions. There's a lot of different things that go on if these things are not working properly. Okay. So that's what I would do in this case here, because you can really go down a rabbit hole with this kind of information. Is that okay? I'm just trying to lay out expectations. So what I would rather have you focus on are the major players and kind of what they do and why they're so cool. Now, what do they say now? 6, 7, K long. Yeah, I know. That's my stupid kids. I've started saying like 8, 9, in order, because they're teenagers. They're funny. My apologies, but yeah, they're still a teenager. So, okay. Yeah, that's okay. I'm old. That's the problem. Like out of it. Okay, let's see here. What else? Just a few more slides. This is oh wow. Antibody dependent. That's really small. I guess that's just kind of the whole overview of what this thing is. So it shows how these cells are made. You don't have to worry about gene rearrangements or anything like that. You have these cells going from the bone marrow. If it's a T cell, it's going to go into the thymus first. And then what do they do after that? They circulate. Remember I mentioned the lymph system last time? So they do a lot of circulation through the lymphatic system. They can go back and forth between lymphatic and the circulatory system. So they monitor both. But then the idea here is that they're going to run into some sort of antigens that are foreign, and then that's going to set off the disease progression, or at least the immune response. Yeah, so that's kind of an overview. I didn't read this case history, sorry, I forgot to read all the way down. So you can read about this, but I think it's basically what I was saying. We can't really be missing anything. Some of the hardest diseases are people that have defects in their immune system, because they usually show up really, really early in life. And these kids are usually really, really sick. So here it looks like lymphocyte is a B and T cell. So maybe this person was not producing antibodies very well, and that's going to be a pretty deadly disease. At the beginning of this slide deck is the case of the bubble boy. You guys ever hear of the bubble boy? It was back in the 70s. That's a really hard existence, because that person basically had no functioning immune system. So they couldn't be exposed to anything without getting seriously ill. Nowadays, there have been some, I'm just going to click through here. You guys can read about it. There has been some gene therapy. I don't know if you guys have ever heard of this, where they've tried to go in and cure SCIDs or something like that, which is severe combined immunodeficiency. And they've had some luck. They've had some luck inserting new genes, which will then fix whatever. It's usually a genetic mutation. And so I don't know if they really take people off of their other treatments, because you don't want them to get sick. But at least there's been some progress. Whereas most of the time, if you're really missing something, it's going to be hard to get over that. Okay. B cells and T cells meet. Hey, man, there's another case study. I didn't realize there were so many case studies in here. Sorry. I think you guys can read about them just as fast as I can. This one is showing a B and T cell. So this is a T cell that's being turned into a T helper cell. So T helper cells, write this on here. Yeah. So cytotoxic cells, they do a lot of killing, basically. The way I think about T helper cells, the easiest way I like to think about it is like B cell activation. So cytotoxic T cells can kill other cells. So if you want to think about like a virally infected cell, we talked about viruses, right? What do viruses do with host cells? Use them to make baby viruses, right? So for the good of the organism, those cells become antigen presenting cells. They basically say, hey, T cell, I'm sick. It's like an alien movie, right? Been taken over by this foreign thing. You better kill me off for the good of the organism. That's what these cells do, at least if it's a multicellular organism. So what are the T cells? The cytotoxic T cells come along, they bind to it and go, okay, sorry, I'm going to release all these compounds and kill you off before all those baby viruses come out of you. So that's what cytotoxic T cells do. B cells, on the other, sorry, helper cells, what do you think they do with B cell activation? They turn the B cells into plasma cells. So T helper cell can help B cells produce more antibody and better antibody as well. I don't know if that's shown on here. This just shows a cytotoxic T cell. So I think that's all you need to remember with those. So two different types of T cells. The cytotoxic ones are the ones that go kill things. The helper cells are going to help other cells like B cells do their job better. And again, the whole goal is to make more antibody, better antibody, get a better response, immune response. So let me ask you another question here. Bless you. So we know kind of what antibodies do. I didn't explicitly talk about this, but I did mention it a little bit. So part of the immune response, making antibodies, making cells that can go kill things, and the other part of adaptive is going to be this. Memory. So here I had to come up with some weird answers again. It's getting late in the semester. My creative juices are running low. You guys get it? Oh, look at that. You didn't, hey, one person said gain superpowers. There you go. I wish. Let me add one thing to this list. Not only does it make a memory, but also it makes better antibodies as well. Keep this in mind as well. Our immune system, even after the first time we are exposed to something new, they can learn to make even better antibodies, which could fight it off even quicker. So, I know it's not, I mean, we weren't able to spend a lot of time in this and show you the nitty gritty, but I hope that you can gain kind of an understanding and an appreciation for all the moving parts in this. Why you should be very thankful if you do have a good immune system. If you have a bad immune system, just call your parents and cuss them out. They're giving you bad genetics, okay? Because a lot of it is genetic. But if you have a good immune system, then we want to take care of it and be thankful for it. Jim.
Yeah, so B cells are antibodies, right? They bind antigens.
B cells make antibodies.
What is it that actually destroys the cells? Is it the B cell?
The cytotoxic T cells can kill things off directly. But if you want to think about something like the antibodies, let me look at that picture again. I can show you that one picture if I can find it. There was one picture that showed how antibodies can bind to things and help them get recognized. Was it over here?
Are they pointing them out and then natural killer cells?
Yeah, right. So antibody can do a lot. I guess that picture was in the other slide. You need to imagine these things can do a lot of things. So they can bind the toxin, and then there's macrophages that will recognize that and kill it off. They can bind the bacteria, and natural killer cells or macrophages can come in and kill them off. They can surround different proteins, and then things like the complement will come in and basically wrap around those and help them get eaten. So macrophages and neutrophils, natural killer cells, they do a lot of the eating, destroying. Antibodies do a lot of the identifying or being like an antitoxin.
Nice label.
Yeah. Or prevent them from binding. So they can bind to a viral particle and keep it from binding to its target. So yeah, I really should, I'll add a slide to this. There should be a list of all the different things that antibodies can do, because there's a whole bunch of things they can do. They all come from B cells. And so that's what's cool about B cells, because then the B cells can switch between these depending on the antigen, whatever it is. Did that answer your question? Yeah, so try to remember the major roles. So B cells make antibodies. What do antibodies do? List down a few things. T cells, you can have a couple different flavors of T cells. There's one, I mean, there's T regulatory cells. Anybody follow the Nobel Prizes? They just won a Nobel Prize about describing what T regulatory cells do. So if you have any interest in allergy or cancer or autoimmunity, you can go work in a lab that studies T regulatory cells, because that's a whole nother class we didn't even talk about. So yeah, so you should know the difference between a cytotoxic T cell and a helper T cell and a B cell and a macrophage, and what does antibodies do? That's kind of the point that I want you to be at. I just mentioned a few. So they can bind to toxins. So it's like a detox. They can bind to viral cells. Remember, viral cells bind to their host protein. Antibodies can interfere with that. Antibodies can coat a bacteria or a toxin or a virus and tell the macrophage to come eat it and the antibody. So the antibody is being digested along with it. That's just a partial list. So you can probably just Google it and find a whole huge list of them. Is that good? Everybody thankful for their immune systems? Okay, good. So remember to sleep, rest, take your vitamins, take care of your immune system. Because when you get stressed out and no sleep, your immune systems crash and you catch everything, okay?